My name's Mark Tracy. I'm not Shanhua Lin. She's the real expert, but I learned everything I know from her. So, charge variant analysis, it's, remember that huge long list of things that FDA wants you to explain about your protein? Charge variant analysis is one of the basic techniques. You've seen these cartoons many times. Lots of different things can change in your protein. It can be because of storage, it can be because of processing, it can be because of the expression system. You get changes in post-translational modifications, clipping of the terminal lysines, deamidations, pyroglutamations, differences in sialylation. It all shows up in the charge of the protein. So lots of different tools, lots of different things you're interested in. We're going to be talking about ion exchange chromatography of proteins today. In this talk, I get to do the next one. So okay, so we've got columns, and I'll get into columns, but a quick detour about HPLC systems. By the way, there's one of them sitting right out there. So, when you get to the break, go out there, take a look at it. A couple of key things about this. The materials of construction is all MP35N super alloy. This is stronger than stainless steel. It's much more corrosion resistant than stainless steel, and it's way more biocompatible than stainless steel. This is the kind of stuff that you could use for surgical implants. There's also this accessory detector, which goes with our Ultimate 3000 systems, and sometime in the not too distant future will be available for the Vanquish systems. This lets you actually see what the pH and conductivity coming out of the column really is. And we'll show you why that's useful to know. Okay, the classic way, of course, is competitive ion exchange. Typically, it'll be cation exchange chromatography with a salt gradient. This is your gold standard method. It's familiar. It's been around. It's easy to make your own mobile phases. So sometime 15, 20 years ago, we introduced a whole series of ProPak columns based on a non-porous polymer bead coated with a very thick layer of hydrophilic epoxy, and then we graft an acrylic material onto that and functionalize the acrylates. So in this way, the protein never has to see that hydrophobic core particle. You get really good kinetics inside the column, even with great big proteins. The performance is really good, so good that at least one of our customers decided to call it the gold standard. And ever since then, we have been shamelessly repeating that quote. So here's a typical kind of chromatogram. Here's our lysine variants. All the lysines removed in this one lysine removed, no lysines removed. Here you have these basic variants. These are more strongly retained. We have our acidic variants. And these variants could be for any number of causes. Now you can treat your Mab with carboxypeptidase B. It takes off those terminal lysines. It collapses these three main peaks into one larger peak. We still have some basic variants. Shanhua, the original author of this slide deck, got curious about what these are. Turns out some of those are actually aggregates. So yeah. Now, we had a great product, but we thought we could do even better. So we spent a lot of time and effort working on something new. And what we got was a strong cation exchange column. And you can see it's got a little bit better resolution, slightly different selectivity. You can get a lot more peaks out of it this way. This is, again, a 60-minute run, so you get more resolution. But since you've got more resolution, maybe you don't need all that resolution. You can save time instead. So go from a 60-minute run down to a 15-minute run. And this is all because of better chemistry. One of the key improvements that we made in this was, this uses the same kind of acrylate-based grafting, but in this case we use a process called ATRP, which is a controlled radical polymerization that lets you get much more exact control over the chain length. So, okay, so a brief diversion. How does salt gradient work? So for simplicity, we will talk about a strong cation exchange. It's got these fixed sites on the stationary phase. We pick a pH for our method where the protein has a net positive charge. In fact, all the variants that we're interested in have a net positive charge, so they bind to the column. And then as the gradient of salt proceeds, more and more sodium ions compete for those fixed ionization sites on the column. And we keep shifting the equilibrium away from the binding. So the interaction happens all the way through the column, and it's in classic chromatography. Longer columns give you higher resolution. Okay, very familiar stuff. But the one big drawback to this technique is you don't know what the right pH is for any given protein. So if you're analyzing lots of different proteins, you have to do an experiment like this where you have a whole bunch of different buffers and you do a bunch of different experiments to determine which pH gives you the best results. This is days of work just to find out that, oh, this protein's not the one we really want to use anyway. So there's another way. And this is the pH gradient technique. So when you do a pH gradient technique, it offers a lot of real advantages. First of all, it's a platform method. What I mean by that is it's one method that works for a very wide variety of proteins. Basically, any protein with an isoelectric point between 6 and 10, you can do this technique. So your method development times are much shorter. Because of the way it works, this is much less sensitive to the effects of the column. It's more driven by the buffers than by the column. So it also makes it very easy to transfer this technique from development to QC. You just buy these concentrates, dilute them down tenfold, and you're ready to go. It works a little differently. In this case, we're starting at a low pH of about 5.2, and at this pH, our protein is protonated, and now we're going to slowly raise the pH, and the protein starts to lose positive charge. Eventually, it becomes neutral, and it unbinds from the column. Now, the neat thing about this is it doesn't matter where on the column that protein is bound. once the right pH passes over the protein, it unbinds and starts to move. So the column length really doesn't have a strong effect on the resolution. So you can get very fast results. Now, one of the problems that you have, of course, is you want a good pH gradient. And there's been a lot of stuff in the literature over the years, but it never really caught on as a popular technique. Well, this is why. Because the pH gradients were lousy. And you wouldn't have known this unless you had that pH detector accessory monitoring the pH coming out of the column. Because all of these literature buffer systems work fine on paper. They work fine in a test tube. But once you put them through an ion exchange column, it's not so fine. And you can see you get these big delta pHs. You get these funny shifts and lumps. Very little nonlinearity when you use the Thermo Scientific CX1 buffer system. And here's a chromatogram. This is one of our quality control chromatograms. And you can see you get lots of peaks, lots of isoforms you can resolve. Phosphate, used in a lot of linear buffer systems, but as you can see, it doesn't work at all well for chromatography. And all of those components that, like down here, the lectins, they all collapse into one peak. All of the little isoform variants of the cytochrome C collapse into one peak. This is not what you want. So here we've shown what the pH is at the elution point. I don't have this figure, but you can actually construct a correlation diagram between elution time and isoelectric point. And so if you have a protein you don't know anything about, after you've run one of these chromatograms, you know what its isoelectric point is. Of course, as you can see, all the action is taking place in a fairly narrow range. So this is how you optimize. You simply zoom in your gradient and expand out your chromatogram. This happens to be a degraded monoclonal antibody. Now, some of our colleagues over in Germany thought, well, we've got this column, we've got these high-speed chromatographic systems. Let's see how hard we can push. So here's a 10-minute gradient, 0 to 100%. Zoom in on the active part. Now it's a 5-minute gradient. Well, keep pushing, keep pushing, keep pushing. The actual separation gradient is just this little section right here at the beginning. It's less than a minute long. We're pushing the column at 1.2 mils a minute. That's fast chromatography. And you'll see the resolution really isn't very different. As I mentioned, this is a platform method. You can do generic chromatography on a lot of different proteins. These are all monoclonal antibodies of one sort or another. These are 10-minute gradients with a little bit of optimization. We now have gradient times below 2.5 minutes. Some of these have cycle times of 7 or 8 minutes. You can get a lot of work done now. Is it reproducible? Yes, it is reproducible. Here's 300 injections. RSD's about 0.8%. One of the non-obvious features of the pH gradient technique is its high loadability. So using the same column, here's the pH gradient on top, here's the salt gradient on the bottom. You can see it's not real obvious, it's very hard to read, but that's 0.1 milligrams, 0.3 milligrams, and 1.2 milligrams. In the pH gradient, you're not degrading the separation at all by loading it. With the salt gradient technique, loading seriously degrades the resolution. Here's a comparison of a 50 millimeter column versus a 250 millimeter column using the pH gradient technique. You have to look really pretty hard to see the differences in resolution. And you can see that, for example, the peak width at half height, it's the same. And why is that? It's because this is driven primarily by the gradient. And so here we have the pH at the elution. There's a bit of a shift, but not too much. Another nice thing about, especially because this is a strong cation exchange column, you don't have to titrate the column when you re-equilibrate it. So it equilibrates very quickly. And you know it's equilibrated because you have your pH and conductivity monitor that tells you that the mobile phase coming out of the column is the same as it was at the beginning. You can use the older ProPak WCX10 columns with this pH gradient technique. It works very well. There are a few changes that I'd like to point out, however. First of all, the WCX column takes longer to re-equilibrate because the column itself is a weak acid and its protonation state can change. Secondly, you'll notice that the gradient is delayed, and consequently, you'll notice that all of the peaks come out a little bit later on the weak cation than they do on the strong cation, and the selectivities are a little bit different. So if you just want to see what this is all about and you don't want to buy a new column, just buy the buffer kit and try it out. One of the things that you probably want to do, of course, is to eventually get some mass spectrometry. So typically what we recommend you do is do fraction collection and then desalting. So here we've used the ion exchange pH gradient. This is the ultimate 3000 LC system. This model of autosampler has an auxiliary valve, and it can actually collect fractions back into the autosampler tray, right into a 96 well plate, and then you simply walk that plate over to your LCMS system, desalt it with the Mab RP column, and get your data. And you can see here you can see the lysine variants, you can see the glycoforms. It's all in there. Mab RP, there's a whole story behind that. I won't go into it, but it's a polymeric material. It's One of its better features is very low carryover. If you've used silica columns, you know that carryover is a real nuisance. This has much lower carryover. Here's some full scan data where we've done this desalting experiment. So I'm not going to try to explain this. This isn't my data. But you deconvolve it. This is what you see. And so here's our various, some of our variants, glycoform variants. And you can see the mass differences corresponding to the lysine truncations. And finally, I wanted to wrap up with some examples. We have a collaboration going with the National Institute of Bioprocessing Research and Training called NIBRT in Dublin, Northern Ireland. And they've done some really cool work, and I'm going to share a few slides of that. First of all, NIST Mab. It's great. It's non-proprietary, so you can use this for your system suitability checks. If there's something not working right and you want to call us up and complain about it, and you can't share your proprietary data, you can share this data with us, and it'll help, diagnose problems. Very reproducible. Reproducibility, even in these rather poorly formed peaks here, is like 0.25%. For these well-formed sharp peaks, 0.05, 0.07. Excellent reproducibility. This is a cetuximab. This is a product that is facing the patent cliff, and the biosimilars are circling around. So here we could have the same sort of thing. Here's some reproducibility data. This is a treatment with carboxypeptidase B. We can see the lysine variants. Same experiment, only this is a biosimilar candidate. And again, reproducibility data, digestion and non-digested. We overlay them, and now, okay, now we can see what's different. And these are the reasons that they're different. They're different expression systems, different conditions. You get different charge variant profiles depending on your expression system, depending on how you use your expression system. So this is all very easy to do now. And this one is the last example. Remember I was talking about how you have great loadability for these columns? Well, in this experiment, we did the folks at NIBRT did cetuximab, and they loaded 50 micrograms on a 2-millimeter column. That's a lot of protein for a little tiny column. And then collected these fractions. So now there's enough that you can digest it with pingase F, release your glycans, and in this case we did it by Heterogeneity chromatography with labeling. And now you've got glycan profiles for each of these variants. And you can see the blue is sialic acid and the gold is galactose. So these refer to the terminal sugars on the glycan structure. So yes, indeed, the early eluting peaks are highly sialylated, and the late eluting peaks are less sialylated, just like you would expect. So I'm not going to bother reading it, but charge variant analysis, MabPAC SCX10, CX1 buffer system, it works wonderfully. And these are some of the people who contributed the data.